Good afternoon, my viewers, my subscribers, and all the people of the internet. Today, I'll take you through a biology tutorial which is very special. 1989 marked the dawn of the 844 system in Kenya. That year, I became an examiner. For the first time, candidates had to prove skills in the laboratory or lab. Below, I'll give the material supplied by KNEC to every school. In question 1, which is dealing with food tests, candidates prepared liquid L from sucrose and formed food tests. In question 2, which was flower dissection, they dissected Crotalaria or any other bean family flower to study its parts. And finally, in question 3, we had the housefly study, where a freshly killed housefly was examined under hand lens. As an examiner, I saw the excitement and the nerves of students facing practicals for the first time in a new education system. 1989 was more than an exam. It was the birth of a system, and I was there. If you sat KCSE in the early 844 years, share your memories below. May I introduce (1989) was a defining year in Kenya's education history. It marked the birth of the 844 system and the first ever KCSE Biology Practical examination. In this video, I take you back to that historic moment, examining the confidential KCSE 1989 Biology Practical paper. The specimens, the reagents, the food tests, and the skills that laid the foundation for how biology practicals are examined up to this day. Because for now, the 844 has reached Form 2, they are facing it out, so that we start with the CBCE. For me, this year holds a special meaning. 1989 is also the year I became a KCSE examiner, witnessing firsthand how practical skills were tested, assessed, and standardized for the very first time. Whether you are a student revising, a teacher preparing learners, or simply curious about history of KCSE biology, this video will give you insight that textbooks rarely capture. Let's step into the laboratory of 1989. Now, let's look at the 1989 confidentials, and we start with question number 1, where each candidate was required to have 10 ml of a liquid labeled L to be prepared from substance L. Substance L was supplied by the Kenya National Examination Council, and the disclosure by the council was that substance L was glucose, or was sucrose, sorry. He also required 6 test tubes, a test tube rack, means of heating or water bath, means of holding test tubes, water, spatula, means of labeling, means of measuring 1 ml volumes, dropper or teat pipette, iodine solution, Benedict solution, dilute hydrochloric acid, sodium hydrogen carbonate powder, 0.1 molar sodium hydroxide solution, 1% copper sulfate solution, Sudan III, and young and green tomato fruit labeled D1, and a ripe tomato fruit labeled D2. Finally, you needed a sharp laser blade or scalpel. And the guidelines for preparation of liquid L, that material which came from the examination council, was you dissolve 20 grams of substance L, which is sucrose, provided in distilled water to form 1 liter of liquid L. Questions now. A. Using the reagents provided, investigate the food substances present in the liquid labeled L and complete the table below. So that was the food tests, and the table is there with the labels, food substance being tested for, procedure, observations, and conclusion. In B, question 1, you are provided with specimens D1 and D2. Cut a transverse section through the middle part of the specimen D1, that is the unripe tomato, add a few drops of iodine solution to the cut surface of the section. Record your observations and conclusion, you get 2 marks. Repeat the procedure in B1 above for specimen D2, that is the ripe tomato. Record observations and conclusions, 2 marks. And Roman 3, explain your observations and conclusions in B1 above. Now, let's come to the answers of question 1. So we go to question 1A, and that is filling the table. So, food substance being tested for, starch. Procedure, to liquid air in a test tube and a few drops of iodine solution. A few drops can be 3 drops. Observation, no color change. Or you can say, takes color of iodine, or you say, becomes brown, because the color of iodine is brown, or becomes yellow, because also sometimes iodine is yellow depending with the concentration. And the conclusion is starch is absent. Reducing sugar to 1 ml of L and about 1 ml of Benedict solution. Heat to boil. Now, you add 1 ml and about 1 ml means you can say to 1 ml of L and an equal amount of Benedict solution. Observation, greenish color. Others would have said blue color of Benedict solution, but accept also no color change. Reducing sugars absent or simple sugar absent is our conclusion. Non-reducing sugar to 1 ml of L and 1 ml of dilute hydrochloric acid, tick. Heat to boil, tick. cool in cold water, neutralize with sodium hydrogen carbonate, and Benedict solution, heat to boil. Observations: green, yellow, and finally orange or brown. Non-reducing sugars present or complex sugar present. Protein: to 1 ml of air and a few drops of dilute sodium hydroxide and then 1% copper 2 sulfate solution. Notice the under there, you start with the sodium hydroxide and not the vice versa. Observation: blue color or purple color and violet color not seen. No color change. Protein absent. And finally, for fat or oils or lipids as food substance being tested, procedure is you add a few drops of solution L and 3 drops of Sudan III and shake or stir and allow to settle. Observations will be evenly colored red solution, and you can accept turns red, where you conclude that fats, oils, or lipids are absent. Let's go to part B of question 1, where we are recording the observations and conclusions after cutting specimen D1 and D2. For D1, the observation after you add iodine solution, you have blue-black color, meaning starch is present. A tick. For D2, you have less intense blue-black or non-blue-black, all patches of blue-black, and conclusion is starch absent. Explain your observations and conclusions in B1 above, and you say the following. During photosynthesis, you get a tick. Manufactured carbohydrates are stored in unripe fruits in the form of starch, a tick. On ripening, tick, this starch is converted into other substances, or you can say simple sugars or carbohydrates, through enzymatic activity, a tick. So 4, maximum 3. May I now go to question number 2 of KCSE 1989, the pioneer class of 844. The confidential was a flower of Crotalaria species, or if the schools miss that, any other range flower of Papilionaceae or bean family labeled D3. And then you are being given a hand lens and a sharp laser blade or a scalpel. You can use a scalpel. Questions for question 2. You are provided with a specimen labeled D3. Part A. Examine the specimen and name the division of the plant kingdom from which the specimen was obtained. Give a reason for your answer. So there you have the division and the reason. B. Using a laser blade or scalpel, cut the specimen longitudinally to obtain 2 identical halves. Examine one of the halves with the help of a hand lens, draw and label the structures observed. You get 9 marks. Part C, describe the floral structures of specimen D3, 3 marks. And finally, explain how specimen D3 is adapted for its pollination. Let's come to the answers for question 2. So, part A, examining the specimen and naming the division of the plant from the plant kingdom from which the specimen was obtained, then giving a reason for the answer. Division is spermatophyta, tick, and the reason is reproductive organs are born in the flowers, or it has presence of flowers. You can just say presence of flowers. In B, drawing and labeling the structures observed after examining one of the halves with the help of a hand lens is our next slide. The drawing, that is at B, and the drawing is going to be marked from the top clockwise. So we have the standard, the keel, wing, stigma, style, anther, filament, staminal sheath, which should have a double line, funicle, ovary, placenta, ovule, flower stalk, receptacle, or torus, or the ramas, sepal, or somebody can say calyx, but you reject any label of corolla. So, how was it marked? We had 3 diagram marks, or drawing marks, and these ones were marked as follows. The mass mark is represented by P, or proportion, where you look at the size, or you evaluate whether the standard, keel, wing are drawn in correct relative size. Also, you check calyx, ovary, and stigma proportions. Also, note the long versus short stalks and their placement. That is the first drawing mark. The second drawing mark is the biological arrangement of the parts of the flower. Here you focus on accurate internal anatomy, that is stigma above placenta, ovules on stalks, collect ovary structure, penalize if placenta or ovules are misplaced or missing. That is the second mark. And the third mark of the drawing is quality of outline. So you require to have clear continuous lines, no shading. Labels must touch the structure, no floating or crossing lines. Check for double lines in staminal sheath, presence of filaments, fused or open calyx. And therefore, the grading notes are as follows. D is 3 marks. That is for proportion, biological arrangement, and quality of outline. Labels, we have 15 labels. You divide by 2. 15 divided by 2 approximately this becomes 7. Okay, so you round down, you don't round up, because you get 7.5. Therefore, the drawing marks are 3 and the labeling marks are 7. The total marks is 10, maximum 9. So we expected, of course, some students to draw various types of diagrams like the one you see here, but you have the rebels again, you have the keel, you have the stigma, you have the filament, you have the style, you have the placenta, you have the stalk, and you have the calyx. So was another type of diagram here, which you could also have marked. Let's now go to part C of question 2. Describing the floral structures of specimen D3. So we can start. It has 5 fused sepals, a tick, 5 petals, a tick, not all fused, a tick, standard 3 wings plus keel fused, or 2 fused to form a keel, or some fused. Petals have different shapes and sizes. You get a mark. Has 10 stamens. 5 stamens have rounded anthers, a tick, while 5 have elongated anthers, a tick. Filaments are fused to form a sheath round the ovary, a tick. 9 fused filaments and 1 separate free, a tick. Monocarpous, 1 pistil or 1 couple, a tick, with about 5 to 10 ovules, a tick. Stigma or anthers is within the petals or keel or corolla, a tick. Stigma is above the stamens, stroke anthers, tick, or anthers are below the stigma, a tick. You get 14 descriptions divided by 2, you get a maximum of 6. Different names given to differentiate accept sizes and shapes. If anther fused with the filaments forming a sheath. If 5 rounded, 5 elongated, accepted for 10 stamens and shape. Reject elongated anthers rounded. D. Explain how specimen D3 is adapted for its pollination. You're having 3 marks. So, this is an insect-pollinated flower. Therefore, has brightly colored, or you can say any other part of petal or corolla, to attract insects. Next, has airly sticky stigma to trap pollen grains, has keel for insects to land on or support, a tick. It has a pistil longer than stamen for cross-pollination or insect-pollinated, a tick. Has nectar guides for guiding insects, tick. Reject honey guides. Pollen guides. All right. All presence of nectaries. Must stress on adaptation, purpose except for nectar guides. If insect pollination not given first, but later, I want the point. Reject if current flower. All right. Note how the parts help in pollination except nectary. Now, let's come to question number 3, confidential. In the first part A, we are the drawings of 4 different animals, K1, K2, K3, and K4. Those were in the question paper. But the teacher who was preparing the practical required also to have a freshly killed housefly labeled M, a ruler, and a hand lens. So let's go to the questions. Question 3. Below are drawings of 4 different animals, K1, K2, K3, and K4. Study the diagrams and answer the questions that follow. So we have the diagrams K1 there, K2, K3, and K4. K1 looks like a crab. K2 is a millipede. We have K3, which is a spider. And I can see K4 is a starfish. Questions 3A. Using observable features in the drawings, state with a reason in each case. Roman 1, the phyla to which K1 and K4 belong, you get 4 marks. So the table there you have the phylum and the reason, K1 and K4. Roman 2, the classes to which K2 and K3 belong. We have class, reason, and then we have K2 and K3. Part B, question 3. You are provided with M. Roman 1, carefully remove one of the wings and measure its length, 1 mark, length in millimeters. Roman 2, with the help of a hand lens, observe and make a drawing of the wing. Label the drawing, 3 marks. Roman 3, work out the magnification of the drawing in 2 above. Right. C, with the help of a hand lens, examine the body of specimen M. 1, state with a reason in each case 2 observable features that enable the specimen to be a disease vector, 2 marks. And Roman 2, state 2 methods that can be used to prevent specimen M from spreading diseases. So let's go to the answers of question 3. And in A Roman 1, the phyla to which K1 and K4 belong. K1 belongs to the phyla Arthropoda. Reason, it has jointed legs or jointed appendages. You can also see jointed ribs. K4 belongs to the phylum Echinodermata. And the reason is the body is divided into 5 arms, or you can call them 5 rays or 5 projections, or you can just say presence of spines or spikes. But you reject radial symmetry. Roman 2, the classes to which K2 and K3 belong. K2 belongs to Myriaponda, or you can call it the Diploponda. Reason is it has more than 8 legs, or many similar segments, or presence of many legs, or 2 pairs of legs per segment. And K3 belongs to Arachninda, and the reason is that it has 2 body regions, or 4 pairs of legs. In question 3b, Roman 1, carefully removing one of the wings and measuring its length. The length ranges from 5 millimeters to 10 millimeters. You get a tick. Roman 2, with the help of a hand lens, observing and making a drawing of the wing and labeling the drawing. So the diagram is there, and this is what examiners were expected to follow. Branches extend from base of two major veins, that is the veins branching, and this checks biological accuracy. Veins should radiate from the wing base. Next, examiners should check whether outline is not broken or overlapping, and this ensures technical neatness and clarity. Then they should check quality of drawing, e.g. correct venation, outline size, and neatness. And this confirms the diagram meets visual and structural standards. Finally, they check the rebels and veins and membranes. There are two, and this confirms correct and complete labeling. So this is the way the total marks was awarded. The drawing mark was 1, and the rebels were 2, and the total marks were 3. To summarize in a table here, D1 means 1 mark for the drawing, and L2 means 2 marks for rebels. So the meaning is that D is diagram quality, and what it assesses is that 1 mark for correct venation, outline size and neatness, no broken overlapping lines, labeling accuracy, 2 marks for correctly identifying veins and membranes. Rainbows must touch the structure. Question 3b, Roman 3, working out the magnification of the drawing in Roman 2 above, to get 2 marks. So the limit of the size expected by the council was 3 to 15 millimeters, and therefore magnification was drawing length of the wing over actual length. So the maximum magnification was times 7. X side must be put. If absent, deny, otherwise drawing accomplished. Reject if units are not correct. Question 3c, Roman 1. Stating with a reason, in each case, 2 observable features that enable the specimen to be a disease vector. 2 marks. The first observable feature is presence of legs that walk on contaminated surface, and we can even put it like legs walk on contaminated surfaces, picking up pathogens. The next observable feature is presence of wings that facilitate movement to and from contaminated surface, which can even be made better by saying wings enable movement between clean and dirty areas, spreading microbes. The next observable feature is hairy body onto which disease microbes attach. In other words, the hairy body traps microbes, not just that. And finally, proboscis feeds on contaminated food. And we can even say it in another way, that proboscis feeds on contaminated food, transferring germs. Accept any 2 points. Reject specks or dirt for germs. Question 3C, Roman 2. State 2 methods that can be used to prevent specimen M from spreading diseases. 2 marks. Covering food. Tick. Proper disposal of rubbish or wastes, or ones to that effect. Eradication of houseflies by use of chemicals or insecticides. Reject spray-alone or pesticides. Poor hygiene or keeping animals clean. Accept destroy breeding places and clean surroundings. Accept any 2 points to earn your 2 marks. To conclude, or as we conclude, it becomes clear that the KCSE Biology Practical of 1989 was not just an examination. It was the foundation of practical biology assessment in Kenya. The food tests, specimen handling, observation skills, and logical recording introduced then remain relevant even today. While equipment and syllabi have evolved, the core scientific thinking has remained the same. Looking back as both an educator and examiner, this practical reminds us that success in biology is not about memorizing answers, but understanding processes, observing carefully, and reasoning scientifically. To students, learn the skill, not just the mark. To teachers, preserve the spirit of practical biology. Thank you for taking this journey through KCSE history with me. If you found this video valuable, like, share, and subscribe, and join me as we continue unpacking biology practical, past,